Hi, I'm Liz Flores Montenegro, and today I'm going to discuss an iPSC-derived human intestinal organoid model of Ebola and Marburg virus infection. Pathogenic filoviruses include Ebola viruses and Marburg viruses, which are enveloped, non-segmented, negative-sense RNA viruses that cause fulminant disease in humans and non-human primates, with the potential of human-to-human transmission, primarily through mucous membrane contact with the bodily fluids of an infected individual. Ebola virus has garnered significant scientific attention and media popularity, while Marburg virus, comparatively, is under-investigated, despite having case fatality rates and symptomatology that are nearly identical to that of Ebola virus disease. Importantly, both of these viruses are lethal, with case fatality rates ranging between 24 and 90%. Presently, there are no reliable models that study the consequences of filovirus infection to the gut, including the gold standard non-human primate model. What I mean by that is that in NHPs and humans, if we compare these symptoms, there is rapid volume depletion, epithelial barrier dysfunction, fatal dehydration, septicemia. There has also been documented ulceration and bacterial translocation that alters the architecture of the GI tract. But there is the absence of vomiting and diarrhea in non-human primates, which is very much present in humans. In fact, the World Health Organization considers vomiting and diarrhea as key indicators of advanced disease. And so these Ebola virus or Marburg virus inflicted patients will lose up to 8 to 10 liters of water depletion. So to better understand what is happening in your gut, and analyze whether this is a direct or secondary effect of the virus, we established a human iPSC-derived human intestinal organoid model, one in which we're able to show stem cells, transient amplifying cells, and then either the cells that make up the absorptive lineage or the cells that make up the secretory lineage. Now we have two distinct organoid types. We have distal organoids, which resemble the colon because they express markers that are present in the colon, or we have proximal organoids that resemble the small intestine because they express markers that are present in the small intestine. In order to achieve these organoids, we use a directed differentiation stepwise protocol. We start at day 0 with human undifferentiated iPSCs, and we move in a stepwise manner to achieve definitive endoderm, hindgut formation, intestinal progenitors. Those progenitors are then seeded in distinct media conditions, and by day 30, we have these three-dimensional spheroids with different architectural morphologies. Now, our single-cell RNA-seq data demonstrates that we're able to capture the multicellularity of the intestinal tracts. So we're able to capture the 5 main intestinal cell types. But in addition to that, we're also able to capture either early, intermediate, or late colonocytes showing this different stages of maturation. In the accompanying dot plot, I am showing canonical markers of those individual cell types. So for example, chromogranin B, chromogranin A, and NeuroD1 are markers of enteroendocrine cells that are expressed in this population and in this population only and less so in the other ones. And here the data is presented in an ascending gradient. So this is all true for each individual cell type. And if we move up here to the top, well, then these markers are present in this particular population, the goblet cell population, and less so or not at all in the others, showing that we're able to capture the multicellularity that is present in the distal gut. Similarly, if we look at proximal organoids, well, the same thing is true. We're able to capture the 5 main intestinal cell types, and in addition, steps of maturation, including either early, intermediate, or mature enterocytes. And in the accompanying dot plot, similarly, markers of either intestinal stem cells, Paneth cells, and the remaining cell types. And so here we're able to capture the multicellularity of the proximal gut. Lastly, we looked at how our iPSC-derived model compares to that of primary gut, primary gastrointestinal tract. And so in your GI tract, well, the majority of those cells are the absorptive cell type, which are the enterocytes in the small intestine or the colonocytes in the colon. Similarly, in our model, and I'm drawing your attention here to the red, we're able to see that the majority of those are the enterocytes in the proximal organoids, or the colonocytes in the distal organoids representing the colon. So now that we have a model that has the cell types that we expect to find in your GI tract, and the same cell proportions, we're able to use that to investigate what these filoviruses do to the gut. Here's a simplistic image of the filovirus structure, the filamentous structure of these viruses. It's 7 genes, rather simple, shown here in this biorender sketch at the bottom. The first thing we did was take a fluorescent Ebo's green virus, one that we're able to visualize so that we can track infection over time and establish our model, establish these time points. So at 1 day post-infection, we're able to see that there are individual Ebo's green columnar base cells in the periphery of all of these organoids that we do not see in the mock infected organoids that had no virus. And as we progress through the infection at day 3, well, then now we see evident viral spread. And by day 7, and I recognize that I'm only showing you distal organoids, well, there is collapse of that spheroid, obliteration of that organoid, and evident cell death. So now we have a time course where we're able to see time points that reflect what occurs in either Ebola or Marburg virus induced or inflicted patients. So if now we remove that fluorescent virus and we take wild type virus at the particular time points, now removing day 7 because there was cell death by then, we're able to see just that at 1 day post-infection for Ebola virus infected organoids or Marburg virus infected organoids, individual small puncta, minimal infection. By day 3, robust infection here in the red for both of these viruses. If we look in at a zoomed in magnified version of one of those cells within the organoids, we can observe infection in the perinuclear region of the cell. We call these inclusion bodies, hallmark of filovirus disease or filovirus infection rather. And we can quantify that. And at 3 days post-infection, about 42% of those cells are infected. Our immunohistochemistry data shows just that. If we look over here at the quantification of that, at 3 days post-infection in the black, there is more infection compared to over here in the gray at 1 day post-infection. Now, if we take bulk RNA-seq to understand what is happening in these organoids, we take our organoids, infect them with virus in triplicates at either 1 or 3 days post-infection, and we can observe that both of these viruses affect the gut similarly at 1 day post-infection. There's an upregulation of hypoxia, there's an upregulation of the epithelial to mesenchymal transition. The cells lose their polarity and adopt this invasive mesenchymal morphology. There's an upregulation of inflammation and of kappa B pathways, glycolysis. There's disruption of the apical surface and junction of these organoids, and an upregulation of apoptosis and downregulation of DNA repair mechanisms. Well, then by day 3, our model is able to capture differences between these viruses, which are rather similar in their evolutionary track. So here we can observe differences in the interferon response. It is being induced by Marburg virus, whereas it is being suppressed by Ebola virus here in the orange. If we look at this a little bit further, Marburg induces a delayed interferon response at day 3 post-infection for Marburg, right, that we do not see over here for Ebola virus. And we can also look at a curated list of genes that are implicated in diarrheal pathologies, inflammation pathologies, irritable bowel disease, all of these diarrheal pathologies, and we can observe that dysregulation between these two viruses in our organoids. Now, if we look at function of these organoids, which is simply 2 things, they either absorb or they secrete, we can use an assay to look at the function of these organoids. We take a compound, that compound activates adenyl cyclase, intercellular cyclic AMP, which activates pKa, phosphorylates the channel, it opens, these ions flush in, and then there's swelling of these organoids. Well, if we look at that in our model, these are our organoid types. Time 0, the organoids look like this. 24 hours later, we can visualize that swelling. Here's a time lapse of the swelling occurring within those hours within that 24-hour time period. Now, if we take that same assay and we introduce virus, what we notice here is that there's a lack of response, which indicates disruption of these ion mechanisms in filovirus-infected distal organoids. And this is important because if we compare proximal and distal, we see that in the distal organoids, there is that absence, that affected response to forskolin stimulation. And this is what has been documented for Ebola virus infected patients, right? And so there was a drug therapy that was used for voluminous diarrhea in Ebola virus disease. And so ultimately, our model represents or mimics what has been documented in these filovirus infected individuals and ultimately serves for translational applications to really study preclinical drug testing and therapeutic evaluation. Thank you very much, and I hope to see you at the conference.